Okay, it's recording again. Yeah, sure, I can begin. And so, as you've seen in the emails, we're fortunate today to have Peter Rogan speaking. Peter Rogan is a professor of biochemistry, computer science, and biomedical engineering at Western. And he's also the Canada Research Chair in Genomic Bioinformatics. And Dr. Rogan is also president of Cytogenomics, which I'm hoping we'll hear more about at the end. So everyone who's joining, welcome, and there was an email sent earlier today with the handouts for the talk. So if you want to check your email, if you would like to have handouts to follow along. And I guess we'll do questions at the end. So Peter, if you want to go ahead, thanks.
Thank you very much, Tracy, and I appreciate the invitation to talk to you. I'm going to present the talk the oral presentation I gave at the hereditary breast cancer meeting last month on VUSs and our approach to this problem. But I'm going to have to turn my volume down so I don't have feedback. Okay. So this is a relatively short talk. I've added a few slides to expand on perhaps some concepts you might not be familiar with. It's definitely going to be a different kind of talk than you're used to hearing. And you might say that it's sort of where the work we do is going as opposed to where it is right now. Some of the tools I'm going to talk about are available through the company we founded. Dr. Nolan and I founded this company, Cytogenomics, a few years ago. We can talk more about the details later, but right now our focus is on making some of the software tools I'm talking about today available to the general community as products. So we're going to talk about VUSs today in heritable breast ovarian cancer, but what I'm actually talking about applies to VUSs in virtually any congenital or somatic disorder, right? So although I'm not talking about it today, we've done a complete analysis of all 445 tumors with clinical information in the Cancer Genome Atlas, and we're working on a paper describing that. We'll be presenting that. Well, we just presented it at AACR, but they use some of the same tools I'm going to talk about today. A couple of years ago, the Canadian Breast Cancer Foundation supported this project that I'm going to talk about, and the basis of the come on, advance. Let's try that. The basis of the project is something you've already seen before. That is that mutation testing in heritable breast cancer, mostly because of ascertainment bias, has found many more mutations in BRCA1 and 2 than in any other gene. But the fact is that a vast majority of the patients that are sequenced end up with VUSs or unrecognized mutations, which we think actually occur either within what's already been sequenced or in untested regions of, in particular, BRCA1 and 2. And the reason why we, well, I'll just come back to this. The reason why we think this is because of a landmark study, Ford et al., senior authors Doug Easton from a number of years ago, where high-risk families with breast cancer were shown to have linkage to BRCA1 or 2 and in 84% of these families. Now, if you look at that paper, the majority of the patients actually were BRCA1 patients, BRCA1 sequenced patients, analyzed patients, as opposed to BRCA2. We think there's a bit of an ascertainment bias, but what it suggested to us was rather than spending lots of effort, though now we are spending some of that effort, on studying sequences of other genes, the so-called BRCA-X genes that many exome studies are pursuing, that what we wanted to really do was look for variation within BRCA1 and BRCA2 that hadn't been recognized as potentially pathogenic. What I need to tell you right now is that at the moment, some of the methods for validating the non-coding variants that I'm going to talk about are very rudimentary, and they're certainly not high throughput. And it is a limitation that everyone recognizes in these analyses. But what I also hope to convince you of is that the vast number of variants that come out of these sequencing studies need to have an intermediate step before functional validation, and that's prioritization, because the problem is much bigger than we thought it was. So, so when we talk about known pathogenic variants, the ones that the large exome studies and complete genome studies tend to focus on, they look at insertions and deletions that result in reading frame shifts, mutations that result in nonsense codons, and a very small subset of the mutations that result in abnormal splicing. That is, those that occur at the dinucleotide sequences at the splice junctions themselves. All of those are thought to produce non-functional proteins, premature truncation and degradation of the protein. Of course, we don't actually have evidence for that, but there's plenty of evidence from prior studies. In fact, I know that the evolution of this field actually started by doing lots of protein truncation tests. So it's fairly safe assumption that in many instances, those types of mutations are accurately called as pathogenic, and they're considered probably pathogenic under the IARC scheme, in some cases actually likely pathogenic if you include multifactorial linkage analysis. However, variants of unknown significance actually represent 80% of BRCA1 and BRCA2 variants, and actually 95% of variants overall. And I'm going to come to a recent paper that Gary Cutting published in the American Journal of Human Genetics, and where he's advocating for this being sort of the main focus of human genetics going forward, and I completely concur. But to do that, we need tools, and we need to recognize that, like in bacterial systems, the same sorts of mutations that affect regulation, stability of RNA, are likely to also affect the regulation of eukaryotic, in particular human genes. It's been well known for many years that splicing mutations are a major cause of Mendelian diseases, and we can talk more about that later. What we do in our work is to basically classify them into subsets, including leaky, inactivating, and cryptic splicing mutations, and I'll talk to you a little bit about some of the tools we've developed for that, which are available to you right now. Transcription factor binding sites are much more complicated to analyze. We believe we've got a fairly good handle on how to go about doing that. The tools for actually validating mutations in transcription factor binding sites are still very low throughput, but I think there's some promising opportunities going forward. And then finally, we need to consider RNA binding protein binding sites. It's well known that these RNA binding proteins, not only do they bind all throughout the RNA sequence, the mRNA sequence, for various things. They are involved in shifting between alternative splice forms. They're involved in stabilizing the RNA so that we don't have the formation of large R loops that can result in nicking and essentially chromosome breakage. They're involved in stabilizing the RNA through binding, especially to sequences in the 5' and 3' UTRs, and of course to the polyadenylation tract, which interacts with the cap site and actually is an early step in determining whether an RNA is going to be translated. So I realize that this is kind of daunting, and many of you may not have considered this before, but I hope you can bear with me and throughout the rest of the talk. I guess I just wanted to point out that one of the sort of hallmarks of this story is FOXA1 mutations, which actually is a transcription factor that binds between the half sites of the estrogen receptor binding site. Of course, many, while not the case very much in hereditary breast cancer, estrogen receptor positive tumors are one of the main classifications. And obviously, a mutation in FOXA1 affects the ability of the estrogen receptor to bind to that site and actually activate transcription. So these are going to be complex events, and unfortunately, we're going to try to present a unified framework that allows us to look at all these different kinds of events, and I'll get to that shortly. So our hypothesis is that non-coding sequence variants include mutations that contribute to the hereditary breast and ovarian cancer phenotype, and our objective in this presentation is to identify and prioritize variants in such patients lacking known coding mutations of the sort that I described earlier. We have, after some effort, an approved research ethics board protocol to recruit up to 650 patients. I don't have funding for that many patients, but so far we've sequenced 7 breast cancer genes, which I'll tell you about in a moment, in 102 patients. We have 48 more under analysis now in a revision of our protocol that includes now 23 different breast cancer, inherited breast cancer genes. The eligibility is shown here, and the key thing is that in Dr. Peter Ainsworth's laboratory, over the last 10 years, many of these patients have been tested, and no deleterious coding mutation was found. And to be fair to him, some of the early testing was simply a protein truncation test. So it's very possible that we'll find mutations he didn't, not because of the lack of sensitivity of his sequencing methods, but because in the time when these patients were previously tested, they were not sequenced. Okay, so the first 7 genes that we analyzed, there's in every case, this is from a review from the large group, William Fowkes group in Montreal, and I have another slide which is a bit more complex. The genes we're sequencing are the ones that are shown with arrows. All of these genes have been found to be mutated in families, obviously to some degree much higher frequencies than others. And looking at coding regions, in particular exons, and as I said, the conserved sequences and the splice junctions. Most of them are involved in double-strand break repair. A couple of them, as you can see, P53, which is high penetrance but low frequency, and ECADHERIN are involved in cell cycle control. So that the first set, the version 1 of our sequencing involved these 7 genes at the top, and our new capture array, which I'll talk about in a second, includes these additional genes. I've highlighted in purple the 3 HNPCC genes, which have been found in individuals with hereditary breast cancer, and I don't know the details, but these genes were selected by the Enigma Consortium, which I'm a member of, and also the Other Genes Committee. There are 2 genes on this list that we failed to include, which we will be including in a subsequent capture array of RAD51C and RAD51D, but we have RAD51B on here as well. The sequencing of the second set is currently being repeated. We had a problem in the second read of our current paired end read of the sequence of this on our Illumina sequencer. But we have, in addition to, well, you'll see what the mutations are, but what I want to get across is these tracks, these black horizontal, black and open areas represent the capture array that we manufacture in our laboratory based on some technology that Dr. Nolan and I have patented. We have an instrument that actually manufactures microarrays that are cleavable, and then we use these microarrays to actually do our own captures. We use a takeoff on the technique that the Broad Institute developed, which involves incorporating a T7 RNA polymerase promoter into the sequence that's being synthesized. So after the capture array is released from the array, we can amplify it by PCR, and then each time we need to do a capture, we simply do an in vitro transcription reaction. So this is a very effective and relatively inexpensive way to sequence a lot of patients with a finite set of genes. What you would notice is I'm showing PALB2 in the top, which some of our colleagues in the Enigma Consortium consider the BRCA3 gene, because there's an accumulating number of mutations that are being found in this gene. And what you can see is that not only are we covering the exons completely, but we're also covering a lot of intronic sequence and sequences both upstream and downstream of this gene. And on the bottom panel, I'm showing you just a sample of the BARD1 gene, where we're covering the regions that are open are actually repetitive sequences, and there's really no point in sequencing those. So while one could use a PCR-based strategy, once you start to get to 23 genes, a PCR-based strategy is really unwieldy, and really the only thing that is both cost-effective and time-effective is a capture array-based strategy. The capture array we make actually covers more than just single copy sequences, which Dr. Knoll and I hold patents on. They also cover some highly divergent repetitive sequences, so we get better coverage than you would get if you purchased a capture array from Illumina. Okay, so just to put the whole set of genes in perspective, and this isn't actually all of them, but if you consider from the paper I just cited, the entire DNA damage response network, all of these arrows correspond to the different genes that we are sequencing. And it's an entirely separate talk to talk about all of them, but the bottom line is that there is evidence for all of them. We've color-coded penetrance. The low to moderate penetrance genes are shown with blue arrows, and so MRE11A, RAD50, and NSP1 are not directly depicted. They're characterized as the MRN complex, but we're sequencing all three of those. Okay, so here's the results from the first 102 samples. And this really kind of blew my mind when we first saw these. We've really carefully analyzed this data. These are, at least 30x coverage and mostly 50x coverage for all of these genes and all of these patients. And what you can see is that exonic variation is a really small fraction of the total variation that we're observing. There's an absolutely astounding amount of variation in introns. There's a fairly substantial amount of variation in the upstream regions containing the promoters, and there's a fairly substantial amount of variation downstream. At the HBOC meeting, just as an aside, where Kathy was there, the fact is that Dr. Livingston presented a new function for BRCA1 involved in RNA pausing, which is essential to prevent chromosome breakage due to excessive R-loop formation. And when he knocked down BRCA1 levels for a variety of target genes, but in particular he used actin, he actually saw that it introduced mutations downstream of the gene, and furthermore, it also turned off expression of the gene. So we need to start thinking not only about promoter regulation, UTR, RNA binding sites, but now we need to start thinking about RNA pausing and the effects of RNA pausing on overall gene expression when, for example, BRCA1 mutations are present in an individual. But the take-home message is that we see a median of 813 variants per individual. And this highlights the issue that is really the main topic of the talk. Is it's way too many for any of us to potentially analyze. Many of these variants have never been seen before, and we absolutely require methods for prioritizing a fairly finite number of variants for functional analysis. So in January, Gary Cutting from Johns Hopkins published this paper, which I guess was a talk that he gave as the president of the ACMGG, in which he proposes that, annotating DNA variants is our next major goal. And in his presentation, he's done this analysis of all the allelic heterogeneity in all the Leiden open variation databases that are curated by the university in Leiden. And what you can see is that there's a large number of the most common genes have perhaps 50 to 75 variants overall in these databases, but and not all of them are pathogenic, of course. So the take-home is that although you can go way out to 8,700 variants or 4,000 to 6,000 variants, maybe those are MHC genes, I don't know what, but the fact is that the number of variants we're observing just in individuals is substantially higher than the number of variants that they're observing in actual databases. So what do we do? So many years ago, I became enamored with, you could say, I really, I've been in bioinformatics for a long time, and a colleague of mine, Dr. Schneider at the NIH, actually proved that you could actually model the affinity of an RNA or a DNA protein binding event using information theory. And this is a mathematical proof. This is not an algorithm. It's not some score. It's an actual proof that relates the Gibbs free energy or the change in affinity due to, in this case, a mutation of a protein for its cognate recognition site. And the thing that's beautiful about this theory is that we can model any protein nucleic binding event. Everything is in common units of bits. And what that means is that when you actually analyze two different proteins that have information contents, one of which is strong and one of which is high and one of which is weak, low, that in fact you can actually infer what the occupancy of those two proteins are on the sequence that they both might recognize. So this is a very strong aspect because it completely simplifies the analysis of gene regulation. Because of the relationship between the second law of thermodynamics, an individual information content change indicates a fold change in binding affinity, which you'll see shortly is actually very helpful for interpreting VUSs. So the first tool that we developed about 10 years ago was the automated splice site analysis server. We had at that point published around 25 papers with others, and I'd gotten so many requests that we built this web server. Last year, we actually introduced a substantial major improvement to that server, which actually allows us to do what we call exon definition analysis. And if we have time at the end, I'll be glad to give you a demo. So we use this tool to predict splicing mutations in both common and rare diseases. There are over 360 citations so far. We analyze private mutations never seen before. It has a demonstrated accuracy not only by ourselves but by other groups for prediction and validation. And it was in the 2005 version of the ACMGG recommendations recommended for rare mutation analysis. And at that point, I went to the FDA and asked what I should do, and they suggested that I submit an application, and they designated this as a medical device. It's not an approved device because I don't quite have the funding to go through the FDA's approval process, but I'm hoping that'll happen someday. Okay, so our approach for analyzing all of this variation is, of course, to do some of the things that you already do in your labs. We use ANNOVAR. We're now using Variant Effect Predictor. We use SNP-Nexus, various softwares that will keep track of how frequent the variants occur in populations, what their effect is on the protein coding sequence, whether they are frame shifts or not, whether they cause nonsense changes. So in that first 102 patients, we identified 9 insertions or deletions, which changed reading frame, 5 nonsense changes. One of these insertion deletions actually started at the very end of the exon and went approximately 120 nucleotides into the intron. And I think it might be missed by some of the conventional approaches that involve just PCR-based amplification of sequences covering exons. We found a number of missense mutations. We ran them through 4 different pieces of software that predict whether something's damaging or not, and only in the cases of 10 of these cases were all of these methods concordant. I am not particularly happy with this approach. I've written a review article about it that was published last November in Human Mutation. I think there are some better approaches, but they're not free. So I myself happen to be enamored with the approach developed by Martin Rees and colleagues at Omisia called VAST, V-A-A-S-T. It's a likelihood-based method. But after we've done what we have to consider, I guess, I wouldn't call it trivial, but it's the coding analysis. we then embark on our information theory-based splicing analysis. We use a tool that we have published and we now make available because we're dealing now with a fairly large number of patients and fairly sizable numbers of variants. It's called the Shannon Pipeline. It's named after Claude Shannon, the inventor of information theory. And it will distinguish mutations that affect natural splice sites from those that affect cryptic splice sites. So we only report the largest changes, so a 4-fold decrease or at least 2-bit change in affinity. And in the case of cryptic splice sites, we only report those cryptic sites whose affinity, whose strength in bits is stronger than the existing natural site. In some cases, they can be pre-existing cryptic sites. In some cases, they can be novel cryptic sites. And in some cases, they can be strengthened by the mutation, simultaneously weakening the natural sites. And after that, we then run the mutations through the automated splice site exon definition analysis, where we can also now look at splicing regulatory sequences such as SRSF1, 2, 3, 4, 5, 6, 10 different splicing regulatory sequences that are known to affect exon inclusion or exon exclusion. Then we use software which we call Mutation Analyzer to analyze transcription factor binding site sequences for mutations. Those sequences, for a long time I held off on building these models, but once the ENCODE project generated a very large amount of positive binding site data, we were able to use software we developed several years ago to build very robust information theory-based models. I'll show you a couple of them later that give us accurate changes in the strengths of transcription factor binding sites. But even then, there are way more transcription factor binding sites in the genome than are probably at all functional. And there's been a lot of work, primarily out of ENCODE, in particular the group from Seattle, that the transcription factor binding sites we need to pay attention to are those that are found in open chromatin. And ENCODE, again, has graciously given us a very nice genome-wide map of DNase hypersensitive regions. So we use that DNase hypersensitivity to filter out those variants that affect transcription factor binding sites that we think are actually relevant. They may be relevant to expression. We report both the changes that strengthen these TFBSs as well as those that substantially weaken them. And then finally, for untranslated regions, we wanted to develop a new approach that combines secondary structure of the RNA with RNA binding protein recognition. So we collaborated with Alain Latterdock, University of North Carolina, who's developed software called mFold that will actually take a large number of variants and then rank them in terms of their propensity to disrupt the folded structure of a secondary structure of the RNA. We then use that as a way to filter those variants that we then evaluate using the information theory-based models derived from Tim Hughes's RNA binding protein database. We can't use all the models that are in there because in many cases there isn't enough data. So we've supplemented those with data that's recently become available through studies of iClip and PARClip, which is basically protein RNA cross-linking experiments analogous to the ChIP-seq experiments to get large sets of positive binding data. And then we actually only we filter basically only those sites that have large changes of at least 4 bits. This is somewhat arbitrary. We haven't optimized these. Again, we're trying to prioritize. Okay, so the results for splicing mutations, these are some of them. There are quite a few more that we find. I'm showing you a number. We found a number of splicing mutations in ATM, and in fact, that's not new. There are reports that ATM is a fairly common target of splicing mutation. I'm going to show in the next slide some results from the CETO that you generate for the first mutation. That mutation is a slightly stronger than average splice donor site or 5' splice site of 10.4 bits. It's completely inactivated. It's a +1 change. Okay, it's somewhat trivial, but not all of them are, because what you can see is that here's the second mutation. While this one is weakened, it actually would be predicted to activate a upstream ATM splice acceptor site at 5.3 bits. So there are a number of examples of pre-existing or strengthened cryptic sites that really would have to be tested in a functional analysis. And it's not so difficult to do that by RT-PCR. But the take-home is that we did find mutations, not just in BRCA, but also in a number of the other genes that have been shown to previously be mutated in families with breast cancer. So this is the result that is shown when you run ACETA. We actually generate these bioglyphs, which not only show the structure of the RNA before and after the mutation, but if there are cryptic sites that are predicted and created, they will also be shown. Each of these are given an isoform number, and in fact, ACETA will also rank order the relative abundance of these different isoforms. So really, you have everything you need to design an RT-PCR experiment to look for particular isoforms. And at some point, when we have time and personnel, we actually will automatically design those primers for you, but we just haven't had the chance. So I mentioned that we had built transcription factor binding site models from ENCODE data. We've actually built 125 unique PWM, that refers to position weight matrices, either information-based weight matrices, and they actually allow us to compute information. So when you look at a sequence logo, and I'm sure many of you have seen these before, you're looking at the average information over all of the sites amongst the set. And when we are looking at the individual information, we're looking at the information of a single site amongst all of those. We can actually back calculate that using these information weight matrices. So here, for example, is an NF-kappa B site. It shows very nice symmetry, and it shows where the main contacts fall. And it turns out that a lot of these sites show very interesting symmetry. So I just pointed this out to say we've automated this process, and given enough data, we can actually build these information weight matrices, and we've done so. Now, after we select these first seven genes that we sequence, it turns out that only 32 of them actually have ChIP-seq peaks in the promoters of those breast cancer genes. So we're not actually scanning, at this point, all of the promoters with all of the position weight matrices that we've derived. Only those where there is recognition and there is DNase hypersensitivity of the transcription factor binding site. So we ended up out of the 102 patients with 84 variants that actually make significant changes, either strengthening or weakening. In these particular case, this is a B-class transcription factor binding site that's very far away from the start of transcription, but it's a rare site, and it actually inactivates that site. But here's one that's actually 54 nucleotides from the start of transcription in E-cadherin, and it actually substantially weakens by 91-fold this FOXM binding site. You can see there are a number of others. CTBP1 and 2 are actually well-known regulators of BRCA1 and BRCA2, and so those are of great interest to us. So here's the FOXM site, and as I mentioned, there's a 91-fold change. FOXM is already known to be degraded in a CDH1 dependent manner. It's part of actually the original MammaPrint paper as one of the genes that is can be used to classify histological grade in ER-positive breast cancers. There's a strong correlation between FOXM expression and the mesenchymal phenotype, and it can be used as a prognostic factor in grade 2 breast cancer patients. So, this is the kind of variant that we would consider worthy of validation and further experimental workup. For the UTR analysis, as I mentioned, we do a two-step approach. The first step is to use mFold to rank variants, and so this is taking all the variants that we find in BRCA2, and we rank them, and in this particular case, we have a variant 52 nucleotides upstream. It has a known RS ID, and it's actually reasonably abundant in the population, but it has a higher propensity, 52 nucleotides into the, it's C dot minus 52. It's 52 nucleotides upstream of the AUG codon. of the RNA. And it's, it has a fairly high p-value. We then took these two here, that this is in the 3' UTR, both of the, and this is in the 5' UTR of adherent and BRCA2, and we looked at whether or not any proteins actually bound to these sequences either before or after the variation occurred. And what we see is that there are some changes. So I'm going to show you the YBX2 and the no-no binding sites that are, in this case is created in TP53. In this case, it's strengthened in BRCA2. There's an HNRMPA1, which is a splicing silencer sequence, which is actually substantially weakened and probably abolished in TP53. So if we just take a look at this BRCA2 C.-26G to A variant, it has a high heterozygosity, but that shouldn't dissuade you. This paper in breast cancer research actually showed that this variant resulted in a 2-fold decrease in expression in BRCA2. And the other interesting thing that we noticed, right, was that when the mutation occurs, it actually exposes this site, this YBX2 site, which is, as I said, 15-fold stronger than the initial site. And what is YBX2? I didn't know anything about it. We have 10 homozygotes and 37 heterozygotes. It's actually expressed in the sperm, but it's also expressed, according to the lunar body map, in ovary and breast. Now, in the sperm, when it binds to an RNA, it actually suppresses expression, translation of that RNA. So you could imagine a mechanism by which YBX2 binding to this variant, this sequence now that it's essentially exposed and available, is actually now preventing the translation of BRCA2. I'm not saying that this is the cause of the mutation, but it seems to me that it's consistent with the results shown in this study that, in fact, the expression of BRCA2 is reduced by 2-fold by the presence of that variant. We actually have been able, Alan, as I said, my collaborator at UNC, has been able to confirm the TP53 UTR variant shown here, which actually had a p-value of 0.07. It has a heterozygosity of 7%. We had 2 homozygotes in our group of patients and 10 heterozygotes. And this particular variant destabilizes a hairpin loop, and that's why it was considered significant based on the method, but that actually exposes a no-no binding site and creates with a 19-fold, 20-fold increase in strength, and it substantially weakens and abolishes this HNRMP1 site. And no-no has a role in DNA double-strand repair. There's not a lot of literature about it. It binds to ADP ribose, promotes non-homologous end joining, inhibits homologous recombination. So, you know, it's, again, a fairly good candidate for a functional analysis. But I just wanted to posit for a second, we could have done the same analysis in reverse order. And if we do it in reverse order, if we, for example, argue that perhaps the protein binds first, and then we see how the RNA will fold, well, in that case, the no-no site is completely open and available. And in the wild-type sequence, it's completely unavailable. The binding site is masked. So at this point in time, we don't know whether to look at the protein binding first or we look at the secondary structure first. But one way or the other, these kinds of differences tell us that they're very good candidates for follow-on functional analysis. So I'm going to summarize all this data. When we look at all the patients and we sum up all the transcription factor binding sites, splice site mutations, UTR variants per patient, and we look at the distributions, what we can see is we end up with on the order of, well, in this case, the median was around 4. Here the median's around 7 for BRCA, 6, 8 variants, 2 variants. PALB2 showed a surprisingly low frequency of mutation, and TP53 also fairly low. And it's not obviously related to the length of the gene because TP53 is much shorter than PALB2. If we just look at the expected number of variants based on length and the observed number of variants, the chi-square is significant for whatever that means. But what I guess I'm trying to point out is starting from, you know, 15,000 variants or 880 variants per gene, we have reduced the number that need to be looked at from a functional point of view to a much more manageable number in the lab. So, and this is, take it for what you think it's worth. It's our approach to variant classification using the IARC criteria. We think the 19 variants are class 4, that we had 2 splicing variants that were class 4. We are not prepared to classify the transcription factor UTR and some of the leaky mutations at this point as likely pathogenic. I still call them uncertain, but I would say with additional experimental evidence, some of these could end up in this category. I don't know how many. But we are able to say that many of the thousands of variants that we did find are probably not pathogenic, at least based on the approaches that we've been using. So to summarize, we have developed a multiplexed automation of sample preparation, which has increased throughput. And what makes our approach different is that we're sequencing the entire gene. We're doing it for a large number of genes using capture arrays that we manufacture here in-house in Canada, not the United States. And we have successfully enriched and indexed sequencing of 7 genes in 102 patients. I've presented a unifying information theoretic framework for splicing, transcription factor, and RNA binding site mutation detection. And I think I've hopefully convinced you that we can stratify prioritize variant lists for future analysis. In my laboratory, most of the sequencing and analysis work have been done by John Mukaki and Natasha Kaminsky. Kobe Viner developed some of the software, and Ben Shirley some of the software we've talked about. Dr. Knowles, an investigator in the project. Peter Ainsworth and Alan Stewart have provided the samples that we've worked with. Karen Panabaker and Abby Watts-Dickens have been painfully and carefully and have been recontacting patients that were studied in the past, and we're potentially interested in talking to some of you if you're interested in participating in our study. It all depends on the number of patients that respond. We've sent out about 400 letters so far, and we've gotten about 100 responses, and we'd like to recruit more patients. And I mentioned Alan and Amanda. This is our support, and I just, again, would like to indicate that cytogenomics is my conflict of interest here, and I'll be glad to take questions. Thank you.
Great. Thank you very much.
I guess I can give the desktop back to you.
Sure. Thanks very much. So if anyone has any questions, we can certainly do them now.
About any particular slides?
Peter, it's Tracy. I did have one question. Like, from your experience with this, do you have a sense of, like, even of the ones you've prioritized as being, you know, maybe you want to follow up with validation experiments, do you have a sense of, like, as you rank them, what, you know, is there a number, is there a certain percentage that you would say fall to the top that you're most interested in?
So I think I want to talk a little bit about what tools are available for validation, just from a...
Sure.
I got to lower you so I don't have feedback. But the tools available for validation, at least on a high throughput level, are, as you know, really not very adequate at the moment.
Yeah.
We have just released a new method, which we call veridical, which means truth. And it is for genome scale splicing mutation validation. And we've applied this to the TCGA data. And it's actually, this paper was published in Faculty 1000 Research. And it's receiving a fair amount of attention. We've given the paper at several meetings. And in the last 2 meetings, we presented at it one best paper. And so it's basically a hypothesis testing based approach using RNA-seq data from the same individual from which you have done an exome or a targeted gene analysis. And essentially, we can end up with a p-value that excludes the likelihood that this isn't alternative splicing or actually normal splicing when, for example, you have exon skipping or you might have a cryptic splice site activation. So we feel that you know, for next generation sequencing, if you have RNA-seq data, and I would strongly encourage you, all of you, to isolate RNA at the time you isolate DNA, because this is coming. I mean, we have the ability on a genome scale to find all the splicing mutations. We have applied this now to the TCGA data set, all the breast cancer. We have found 1,000 or 1,100 splicing mutations that we can confirm, which is twice as many as TCGA reported in their Nature paper. So I'm pretty confident that from a validation point of view, we've got the splicing mutation problem solved. Now, for transcription factor binding sites and RNA binding proteins, at this point, you know, the tools are simply not high throughput. We have some ideas about how to do this. I've got some we're going to propose a CFI application soon, which we hope you don't strike down, which will use mass spectrometry to look at protein nucleic acid interaction and specifically to look at wild type versus mutations. And this will be a moderate to high throughput method, which we think will be a way to actually prove that some of the variants we claim are affecting transcription factor binding really do. I mean, we were fairly stringent in our criteria by saying we insisted on changes of at least 4 bits. It's difficult to say when you create a new transcription factor binding site whether that will be pathogenic or not. Certainly, when we strengthen a splice site, we have, you know, our earliest papers have shown that those are polymorphisms. So, but the question is, when you basically endow a promoter of a gene with a new transcription factor binding capability, are you changing the regulation of the gene and how? So I probably would, at least on the first pass, put aside any transcription factor binding site mutations that strengthen a binding site. Does that kind of answer your question?
Yeah, absolutely. Yeah, I think that's a great point about the need for the high throughput tools too.
Well, we've definitely solved the splicing problem, but you need RNA-seq. And RNA-seq is relatively inexpensive these days.
Yeah. Are there any other questions?
Peter, you offered to, Stacy from Edmonton, you offered to give us a brief preview of, I think, the setup, Spida.
I will give you a couple. I will give you a couple of demos. If there are no other questions, I'll be glad to give them. No more?
Sounds good.
Did I just confuse everybody?
In a good way.
All right. Well, here we go. All right. So I have. No, not it. I have. Okay. So we have this tool called the Shannon pipeline. Initially, we produced it for the CLC biosystem. It's a plug-in for the CLC biosystem. And the problem was the cost of developing it for that system, I think, made it a little bit prohibitive for many labs. The National Cancer Institute did buy it, and they are using it, and they're finding lots of mutations that they didn't know about in their exome data. But we figured out a way to make it more cost-effective for everybody else. And what we do is we set up subscriptions that are term limited. And the reason we're doing it this way is, you know, I run a lab too, and I understand that often the data that we collect is bursty. What that means is, you know, we have a set of patients, we analyze them, and we need to analyze all that data all at once. It's not like, you know, we have a little bit of data today and a little bit tomorrow. We have a pile of data and we want to analyze it all at once. So by setting up term-limited subscriptions, we are able to reduce the cost pretty significantly. So we have a video on the website. This is the Shannon pipeline. So we've moved it now onto the internet. It's a web service, and you don't have to upload any patient identifiers. You can give it anonymous identifiers if you want to keep track of different patients. It uses ordinary VCF format that comes off of most of your, you know, MySeqs or ion torrents, or standard variant call format. And so if I actually, so there's some videos that Ben Shirley, the developer, has put on the website as to how to use it. There's also the manuscript we published last year. in genomics, proteomics, and bioinformatics, which is called Interpretation, Stratification, Evidence for Sequence Variants Affecting mRNA Splicing. We analyzed 3 different cancer cell lines, and we had RNA-seq data, and this data came from the Science for Life Institute by the Karolinska in Sweden, some collaborators there. And so if I sign in, this is the screen you get, and we actually set this up as a queued system. It's really fast. So we have built-in filters. You can start with the ones that are already here, but you can, of course, change them. So, and all the descriptions of any of these things are just by putting your cursor over these eyes, you get a tooltip that tells you what, in fact, the filter is doing. In this case, we've set it to display mutations on both strands. If genes are in the data set that are from both strands, display both donors and acceptors. We can hide variants with average heterozygosity based on frequency. We can hide cryptic sites that are basically have decreased RI. We can hide cryptic sites that have weaker binding sites than the natural site, et cetera, et cetera. I have 2 sample runs that we can run. One takes less than a minute, maybe even half a minute now, and the other takes about 5 minutes. And what happens is, so I'll run this one. You can upload, as I said, your VCF. Here's a video right here on job submission. So if I do run the sample run, it'll use these criteria, and it just submits it to a queue. And the queue depends on how many variants are submitted. So you can technically take all the patients you want to study, give them each a different identifier in your VCF file, and run them all at once, or you can run them at different times. It's your choice. So when this runs, you can see there's a light bulb comes on. It says that our job is currently executing, and you get notified when it's complete. And so I now go to another screen, and you can see that job is finished, and here are the results. Now, we can look at the run statistics. There were 5,000 variants tested. Just to give you some idea, when we benchmark this, we can do 5 million variants in about 10 minutes. It's really quite a different beast from the automated splice site analysis server. We can go back, and you can download the results unfiltered and filter it yourself in Excel, and we give some examples of how we do that in our manuscript. You can download the filtered data, or you can look at it on the screen. So let's just look at the inactivating mutations. And so in this 500 set, which came from, these came from, by the way, the TCGA tumor data. So they're all somatic mutations. Here's a mutation that inactivates a BRCA1 site. It's a known RS ID, but it has a 0% heterozygosity, so it's essentially very rare in the population. It's the coordinate. This is the input ID we gave it. As again, you can make that. It's a single nucleotide variant. The standard pipeline will handle both single nucleotide and insertions and deletions that alter mRNA splicing. And it tells you what the change in RI is. If you have a very large list, like here's the number of cryptic sites, which is there are 29 entries, but you know, there could be hundreds or thousands. You can actually sort these just by clicking. So if I sort by final RI, it sorts. Now you can see an increasing value, and now if I sort here, in this case, this is a variant that actually strengthened a cryptic splice site in this gene LMNB1, laminin-B1. It's intronic. It's the nearest existing natural site is weaker than it. It's 11 bits. This is 53 nucleotides away. And it tells you that this is greater versus lesser. You can, of course, sort. You can sort on this as well. Okay. So we've separated. So leaky mutations are those where there's at least a 1-bit change, a 2-fold change in binding, and they're not mutations that inactivate, but they would reduce the expression. And if you want more information about these, some of my earlier publications talk about leaky mutations. I can give you, you can contact me privately and I'll give you that information. But this is exactly how it works. You can, you know, if you have multiple people using this thing, then you know, everything will get queued up, but because it takes such a short amount of time to run this, the pipeline, you know, it really isn't, you don't have to wait very long for the results. What else was I going to say about this? So, yeah. Okay, so, oh yes, if you sign up for a trial, you can upload your own VCF file, but it will limit the output to 500 variants, and it will only provide you with a small fraction of the total of inactivating variants, because the idea is for you to see how the thing works in a trial, not to do your clinical and research analysis, you know, because it's cost us a fair amount of money to develop this. So, you know, as I said, the cost is relatively inexpensive. Now I'm going to show you the automated splice site and exon definition analysis server. Again, we have a tutorial video. There is a link to the new paper on vertical here, which you can actually go to. This is the main page. Right now, the site is hosted at Western, but it's going to be very shortly going to be hosted at Cytogenomics. Just to give you some idea what our plans are, there are a lot of companies in the genome interpretation field that feel that they need to provide a one-stop shop for all of your needs, and we are going to do that too. We have trademarked the name Mutation Forecaster, and we are going to include an LOVD database, the variant effect predictor, as well as the UCS, the genome browser that Cytogenomics has that contains basically tracks that show all of our probes, our patented probes for both fish and for oligonucleotides for either microarray or NGS capture. So that will all be a single application sometime later in the summer or in the fall. But now we're going to run the ACETA server, and this is a familiar interface for many of you who've actually used it before. I'm just going to run the sample mutation, which is a simple BRCA1+1 mutation. We're taking a wider window range because there are splicing cryptic splice sites that get activated outside of exons within introns. Now, we've changed this interface a little bit so that you can select amongst the different binding sites that you want to evaluate. Our classic acceptor and donor sites from our original 1998 paper are shown here. We have a genome scale and donor and acceptor site for as well based on our 2003 paper. There's a branch point, and then there are a bunch of different splicing regulatory sequences that you can evaluate for their effects. If you see our paper last year in human mutation, we give some examples of these. So I'm just going to not select any of those. We're going to run a point mutation analysis, and we're going to run molecular phenotype prediction. So the first thing it asks you is which isoform. The isoform that Enigma uses is this one, NM007297. You have a number of options. You can translate all frames. You can calculate a standard error on the information content. You can include the effects of a particular splicing regulatory sequence, if you like, and you can indicate how many bits are necessary for an exon skipping event. Then I submit. It generates this metadata, which you can download, and in about a minute, it will analyze that mutation. If you be patient with me for a moment, you can see we're looking at genome build HD19. It tells you all the stuff you need to know about what's being analyzed. And when it's done, it produces two tabs. One is the point mutation, which is the original ASA, and you can see that this is a mutation that inactivates a spliced donor site. It goes from 7.5 bits to 5.3 bits, and this is color-coded, showing that it's abolished. But you can see there are a number of other potential cryptic sites. And we wanted to find a way to improve this, but before I do that, I'm just going to show you the sequence walker. And this is a method that allows us to display these results. So here is the spliced donor mutation. Right here at the end of the arrow, and you can see that we start with this 7.5-bit donor site. And after the mutation, the other end of the arrow shows us that that mutation is now minus 5.3 bits because this is an unfavorable base. and it also shows us a number of other sites in the vicinity. To make this a bit simpler, I guess, we actually can compute the total information across an exon, which is what we term exon definition, and that allows us to compare the relative strengths of exons with the potential cryptic exons that might form. And what you can see is that before we start this, the initial total exon information is 13.9 bits. After the exon is mutated, it's 3.9 bits. Before, there's a 2.6-bit site, but afterwards, there's an 8.1-bit site. This is exon. This is not a very strong exon if you look at our manuscript. And so even though it's a potential cryptic exon, it's actually lower than the average strength exon. And so we can look at the isoform structure. So as I said, here's the starting mutation. Here's the skipped exon. These are the other alternatives that are weaker than even the starting mutation. The starting exon, the wild-type exon. We can look at the relative abundance by comparing these different isoforms according to the numbers here. And this is a 3D plot that does that. I don't really have time, I think, getting into it. And then we also generate a genome browser track, if you like that sort of thing, which shows all of these different potential cryptic isoforms relative to the known exon in the BRCA1 gene in this case. And you can turn on whatever tracks are off you'd like in the UCSC annotation. We've color-coded this using the color spectrum. The strongest exon strengths are red, and the weakest are black in this case. And so that's, you can download the metadata, or you can do a new mutation entry. All the models are here of all the different sites that you're analyzing. All the references that have cited our work, these are the ones that we've published, but there are also citations in the literature, and this is not even complete anymore, of all the studies over the course of the last 10 years that have used the server. So feel free to contact me if you have questions about it. I guess that's it.
Great. Thank you, Peter.
Cost for that one, Peter?
This one we're asking for $200 per year, but we are intending to bundle. We're intending to bundle everything into one, resource. But again, our goal was to try to make it available broadly, as opposed to, to maximize the revenue. As much as I can tell you that we have been able to get subscriptions from around the world. It's certainly not enough to sustain our company. so I don't know quite what this. Go ahead.
I said 200 a year is pretty reasonable. We can afford that.
Yeah, that's right. So my preference would be to have lots of people use it. Sort of the Coca-Cola model rather than the aluminum model. I guess, I personally have a big objection to spending thousands of dollars to buy DNA polymerase and nucleotides per run.
Yeah.
And yet at the end you end up with all this variation, and you're confronted with how do we analyze it, and somehow the community seemed to get the idea that we should just give all this away. But when I have to hire a professional software developer, I can no longer do that.
Great. Thank you very much, Peter. Maybe in the interest of time, if people have follow-up questions, would it be okay, Peter, if we contact you via email?
Yes, it would.
Great. Thank you. And just for everyone, as a reminder, the next call will be Thursday, June the 26th at the same time at 2 p.m. And you can expect some emails from us the week before. Thank you very much again, Peter.
You're welcome, and good luck with your research or your clinical work. Bye-bye.
Thank you. Bye-bye. Peter, I'll send you the recording.
Okay, thank you. Oh,
Yeah.
you know, I assume it's a link, right? Or is it an actual file?
I have to double check that, but I'll figure it out. I'll email you about it.
Okay, if it's a file, then you can just upload it to YouTube and let me know what